Good
Okay.
afternoon, everyone.
Yes. I will try to speak in Portuguese and English at the same time. So, in the first moment, thank you to come, Caesar. It's a big pleasure to have you here. I'm not so happy because you are not really in Brazil, and I'll be much more happy if you are here, and then you can see your talk. I can see you personally. Then you can have some beers after your talk. So, thanks a lot. I will just give the words to Flavia. Flavia will introduce you, and then we can have all your knowledge. Thanks a lot, man.
Thank you, Octavio.
Thank you, Professor Oct. Good afternoon, everyone. My name is Flavia. I am a student of master's degree in biotechnology at Católica Dom Bosco University, and I will be with Dr. Otávio as a moderator. First, I would like to thank you, Professor César de la Fuente Nunes, for his presence. It's a great pleasure to have you with us. I will make a quick introduction about you, and I will give you the floor. After the lecture, we will open for questions, okay?
Great.
Pessoal, para todos que estão ouvindo a gente, eu vou fazer uma introdução bem rápida sobre o professor, e após a nossa palestra, a gente vai abrir para as perguntas, e caso alguém tenha uma pergunta, pode deixar no chat em português, que a gente mesmo traduz para o professor, ok? So, César de la Fuente is a presidential assistant professor at University of Pennsylvania, USA, where he leads the machine biology group, whose goal is combining the power of machines and biology to help prevent, detect, and treat infectious diseases. Current application areas in his lab include developing novel approaches for antibiotic discovery, building tools for microbial engineering, and creating low-cost diagnosis. Specifically, he's pioneered the development of the world's first antibiotic designed by a computer with efficacy in animals model. In addition, he designed pattern recognition algorithms for antibiotic discovery, successfully reprogrammed venoms into novel antimicrobials, create novel resistant-proof antimicrobial materials, and innovate rapid low-cost diagnosis for COVID-19 and other infectious diseases. Professor César has received over 50 awards. He was recognized by MIT Technology Review in 2019 as one of the world's top innovators for digitizing evolution to make better antibiotics. His scientific discoveries have yielded around 100 peer-reviewed publications, including papers in Nature Biomedical Engineering, Nature Communications, PNAS, ACS Nano, Cell, Nature Chemical Biology, Nature Communications, Biology, Advanced Materials, and multiple patents. Again, Professor, I would like to thank you for the presence and the availability. Now I will pass the floor to you, and thanks again, and feel free to start.
Great. Thank you so much for the kind introduction, both of you. And I just want to say that it's really a pleasure to be here, even though I'm very, very far away physically. But hopefully soon I can make a visit to Brazil and to the university, and we can meet in person and talk science in person. So today what I thought I would do is tell you a little bit about what we've been doing the last several years, both in my lab and with collaborators, including Octavio's lab. So one of the things that we have observed, right, in the last many decades is two major trends. The first one, showing on the left here of the slide, is that it has become, it takes a long time to develop drugs, right? So on average, it takes over 10 years to take a drug from the laboratory when you start working with it, all the way to the clinic where patients, people can actually benefit from the effects of the drug. So it's a long, windy road. And at the same time, it's also incredibly expensive to develop a drug. That's what I'm showing on the right of the slide. So basically, one of the latest estimations is that it takes it costs over $2 billion with a B to develop a drug. And just to put this into perspective, this is more than the budget than the budget NASA has to take a rocket all the way to the moon. So developing a drug is actually a more costly process than taking a rocket all the way to the moon. And that puts things into perspective. And in my lab, we're interested in two other trends that we think can help, mitigate some of these issues. The first one is that we have more and more compute power following Moore's law. Over many, many decades, our ability to compute information has increased enormously. And at the same time, we also have more and more capacity to generate data, right? Biological data, chemical data. And so what my lab is interested in is combining these two trends so that we can try to decrease the time that it takes to discover a new drug, and also try to decrease the cost that is associated with developing new drugs. And in particular, we're interested in not just any drugs, but initially we're interested in antibiotic discovery. And the reason for this is that antibiotic-resistant infections are a huge problem worldwide. The latest projections is that by the year 2050, if we don't develop any new therapies, around 10 million people will die in the world every year as a result of these infections. As we can see here, this will surpass every other major cause of death in our society, including cancer, as we can see here. And if you run a quick calculation, those 10 million deaths per year correspond to one death every 3 seconds. And so this is the future that we're heading towards unless we do something about it. And to really put into perspective the importance of antibiotics, I'd like to highlight a couple of things. So antibiotics are not only useful to treat infections when we get infected with a particular pathogen, but they're also essential for modern medicine. So to perform contemporary medicine as we know it, antibiotics play crucial roles in many, many interventions. For example, they're crucial for childbirth. They're crucial for chemotherapy treatment for cancer patients. They play a big role in organ transplantation, and they're also really important in surgeries, any kind of surgery, even a minor surgery like a C-section. You need antibiotics to sterilize the surrounding area. And so they really play a huge role in modern medicine. Without them, we wouldn't be where we are today. And for many, many years, we have relied on nature to provide us with good antibiotic molecules that worked against bacteria. And that all started with the very first antibiotic that was ever discovered, which was penicillin, which was discovered by Alexander Fleming. And that was in 1928. And every other discovery subsequent to that has also primarily been derived from the natural world. And here I'm showing some of the chemistries that biology has produced for us throughout the evolutionary process that have allowed us to use these molecules as antibiotics, right, to treat otherwise deadly infections. The problem is that for a number of decades now, the scientific community, we have been unable to find any new classes, truly new classes of antibiotics in nature. And so our reasoning here is that, instead of relying on nature to be a source of, new drugs, why not try to encode all that chemical information and translate all those chemical bonds into ones and zeros, so then computers can actually take care of the discovery process. And in particular, a lot of our work focuses on a family of very small proteins called host defense peptides, or HDPs for short. These are conserved throughout the tree of life. So bacteria produce them, insects. We humans produce them as part of our innate immune system. Actually, these HDPs, these molecules, they allow us to protect ourselves from otherwise invading organisms that can be lethal. They're typically very small, the molecules we work with, between 10 and 50 amino acids in length. which makes them chemically synthesizable using solid phase chemical synthesis, and we can manipulate these things in the laboratory. But how do we engineer these molecules? Well, traditionally we have used principles from, classical principles from protein engineering that tell us that the structure or sequence of amino acids, which are the building blocks of peptides and proteins, determines their biological function. And so this basically tells us that if you can control the sequence of amino acids, you'll be able to control the biological function of these molecules. And the analogy that I like to use here is that it's almost like playing Lego with these molecules. We're trying to rearrange the amino acids in different positions to be able to, in the end, generate a molecule that, in this case, targets bacteria and that we want to target. But in addition to these more traditional methods or principles, more recently, we have started to incorporate other principles into how we reprogram these peptide molecules. And these are principles not only from chemistry, but also from physics, from computer science, from synthetic chemistry, etc. And the way we like to think about it is that first we want to really understand these peptides, right? How they operate, in terms of structure-function relationships. Once we've reached that level of understanding, we can control them in laboratory, both chemically and biologically, in terms of being able to mutate them and manipulate them in sort of predictable ways. And once we've reached that level of control, the next step is to try to transfer all that knowledge onto computers. And that's what I'd like to focus on today, that latter part of our approach and our framework. And there are two main things that you can do with computers. One is you can explore sequence space, and two, you can try to generate new molecules. And so I'll talk first about how do you explore sequence space. And again, this is incredibly difficult. And just to illustrate the complexity, I'm going to show you a slide with different combinatorial sequence spaces of different things, perhaps concepts that we're all more familiar with. So the number of people on Earth is around 10 to the 10. The number of sandcorns on Earth is 10 to the 28. The number of stars in the universe is 10 to the 31. And if we take a very small peptide composed of only 25 amino acids, one next to each other in a linear chain, again, this is much smaller than any protein that is in our bodies, which is typically composed of hundreds of amino acids. Well, that will give us a sequence space, that very small peptide, that is superior to the number of stars in the universe. So clearly, we need computers to help us with this huge problem. To make things more complex and therefore more interesting as scientists, biological evolution through the evolutionary process has only sampled a tiny fraction of the entire space of possibilities of all potential molecules. And that is depicted by all this white space. This white space has never been explored before. And so another thing computers can do is they can expand the boundaries of sequence space to try to reach these white areas that may give us new sequences of molecules that could serve to solve present-day problems like antibiotic resistance. And I'm going to tell you about a couple of things that we've done in this regard. The first one, was actually a collaboration with Octavio's lab, where we decided to innovate antibiotics using principles from evolution themselves, from Charles Darwin, the theory of natural selection. And this is how we did it. We started with an initial population of the peptides from nature, and then we essentially performed, through mutation, selection, recombination, evolution in silico. Here's just one example. You can see that upon different iterations, we get increased fitness value, which correlates with predicted antibiotic activity. And at the same time, the molecules that the algorithm is generating are different from in terms of amino acid ratio. They're different from what we typically find in nature. So here I'm showing some of the molecules that the computer created at this point. And but of course, this is only a prediction, right? We still need to validate a lot of this. So what we did here is we synthesized some of these molecules, and then we validated them in vitro against actual bacteria. We're able to find a lead molecule that had a very low MIC, minimal inhibitory concentration. What that means is that you need a very low dose of the molecule to target bacteria. And that's what you want, right? You want it to be very potent. And this is the three-dimensional structure of the lead peptide, as elucidated by nuclear magnetic resonance. And here's the same structure of the same molecule in three-dimensional space. And the next step here was we wanted to see if this while binding to peptide was also capable of targeting infections in an animal model, in a realistic animal model, right? And so we infected mice. This is a skin infection model, and we treated them with these while binding to peptide, and we can see that the treatment was capable of reducing the infection more effectively than 2 of the templates that we started the optimization process with. And here we have the untreated control group of mice, so you can get a sense of how many bacteria are in the mice that are left, without treatment. So that's just one example of a project that we completed. We've also since also used other tools to find patterns that have interesting properties. So here we found this. Again, this is also another collaboration with Octavio's lab. We've had many fruitful collaborations, many projects that have led to papers over the years. And here we found this pentapeptide motif, colored in red here, that was associated with antimicrobial and immunomodulatory properties. And so what we did is we took a toxin from a venom, and we incorporated that pentapeptide motif onto the N-terminus of the venom, generating this synthetic version of the peptide. And then we were able to demonstrate that the synthetic version was capable of targeting infections not only through the classical method of targeting the bacterium and killing it, but also indirectly by boosting the host's own immune system to resolve the infection. And we demonstrated this in vitro and in animal models, as we can see here, and also in a sepsis infection model. Again, I'd like to highlight here that sepsis is a huge problem worldwide. It kills 11 million people per year. It's not only caused by bacteria, in the context, for example, of COVID. A lot of patients with COVID, through the viral infection, they eventually can develop sepsis, and which can be lethal very, very rapidly. And in fact, it's estimated that around 1 out of 7 COVID patients also gets infected with secondary bacterial infections, which can also lead to sepsis. And so sepsis takes on many forms and affects many, many individuals every single year, like I'm mentioning here on the slide. But the bottom line out of these experiments is that the treatment with the synthetic immunomodulatory molecule was able to confer protection to mice against otherwise lethal laboratory bacteria strains and also antibiotic resistant strains from hospitals. So that was quite exciting to see. So in addition to using computational tools coupled with experiments to design new antibiotics, we're also interested in using computers to be able to discover novel classes of antibiotics, perhaps. And the truth is that there's a lot of information out there in terms of genomic and proteomic information that has not been properly explored previously. And the way I like to think about this is a little bit like the junk DNA concept, that for many, many years we thought junk DNA was really not doing anything. It was just there. But since then, we've learned that junk DNA has a lot of cellular, a lot of roles in the normal functioning of organisms. And so in order to address this and to be able to mine biological information from novel antibiotics, we took inspiration from image recognition and speech recognition algorithms, but instead of recognizing facial expressions or sounds, we wanted to recognize molecular patterns that were associated with things that we knew could potentially be antibiotics. And the analogy here would be like if you have a huge Word document, like hundreds of pages, and you wanted to find the word hello within the document, you would go to the search function, you would type HELLO, and the software would find the word hello in every instance that it would appear throughout the page, the 400 pages. So this is essentially how these algorithms work. You can have, for example, an entire protein depicted in 3 dimensions. You can then display it in 2 dimensions. The algorithm runs through the code of amino acids, and it finds regions that are predicted to be highly antimicrobial. And these are given by this antimicrobial score and depicted here by the different colors. Again, only a prediction, because it's only the algorithm predicting that these regions might be useful, but it's a very powerful one because it allows us to take fragments from entire proteins, such as this fragment here in yellow, that may constitute a novel antibiotic or a novel drug, and then we can take them from the molecule, we can extract them, and we can play around with them in the laboratory to try to develop them into novel drugs. So what we've done is we've applied these algorithms to explore the whole human body as a potential source of antibiotics. And the interesting thing is that we found thousands of new what we call encrypted antibiotics, that they're encrypted in larger proteins, that a lot of them were thought to not do anything, not have any sort of cellular roles. And the interesting thing is that they're not only produced by the innate immune system, which is where you would imagine you would find antibiotics being produced in our bodies, right? Because the innate immune system is the system within our bodies responsible for fighting off invading pathogens, right? But we also found them in the cardiovascular system, digestive system, nervous system, etc., telling us that host immunity may not necessarily just be the domain of innate immunity, but there may be other body systems within our bodies that are also contributing actively to immunological response. So we took some of these encrypted molecules and we synthesized them in the laboratory, and then we tested them for anti-infective efficacy. We demonstrated antimicrobial activity in vivo in a mouse model, both in monotherapy and also in combination therapy. So if you combine different encrypted antibiotics, they synergize between them to treat infections caused by different pathogenic organisms. Other interesting properties that this encrypted antibiotics had is that if you take some of them from the same biogeographical area within the body, they actually tend to synergize with each other to target pathogens. So they potentiate each other's activity and ability to eradicate pathogenic organisms. And this we saw through synergistic interaction assays. Here, this is actually a 3-way synergistic interaction assay where you combine cocktails of up to 3 different peptides, and you see how they potentiate each other's activity. And what we saw is that they could actually kill microbes when using combination at nanomolar levels. So these are extremely low quantities of peptide needed to actually exhibit antimicrobial activity. Another interesting property that we observed is that bacteria have a hard time developing resistance to these encrypted antibiotics. So here we performed this longitudinal study, and over 30 days, we exposed Acinetobacter baumannii. This is a gram-negative bacterium. It's actually also known as the Iraqi bug because it typically infects soldiers in the battlefield in Iraq, and it infiltrates into their wounds, and it's very difficult to treat, very recalcitrant to conventional antibiotics. And so we can see that the bacterium eventually develops resistance to a classical antibiotic that is used in the clinic, in the hospitals, called polymyxin B, but it doesn't develop resistance to 3 of the encrypted antibiotics that we exposed them to. This slide is just to illustrate that these are just 4 examples of 4 encrypted molecules that are actually produced physiologically in the body in different biogeographical areas or different areas of the body, as we can see, and at different levels, from low to medium to high expression levels within the body. wanted to mention also that we're also interested in developing materials that are antimicrobial, particularly interesting in a pandemic time where high touch areas are problematic, like things like a computer keyboard, or a doorknob, or a surgical table. You want to make sure that the surgical table before you operate on somebody is extremely clean and it's not infected with anything. So we're interested in developing materials that are highly antimicrobial, and we developed these ionic liquid based materials that bacteria do not develop resistance to them. And the interesting thing in terms of application is that you can use them to coat surfaces, to generate antimicrobial surfaces. And you can do them you can coat glass materials, you can coat plastic, you can coat medical catheters. Medical catheters, one of the main problems they have is that when they get introduced into the patient, oftentimes they get infected, and then the patient has to undergo surgery to replace the catheter. This is also always hugely inconvenient, costly, and could actually be dangerous. And so why not try to create a catheter that is antimicrobial and prevents bacterial colonization? And that's what we did here, and we demonstrated here in this paper. But just to sort of wrap up this first part of the talk, we are interested in using computers to both design and discover novel antibiotics. And what we're working on now is coupling that with a robot that will synthesize, that will make the molecules the computer tells it to make. And then we're looking to streamline that further with another robot that will screen these peptides or other molecules for antimicrobial activity. These are functional assays. Again, we focus mostly on antimicrobial activity, but you could think also of anti-cancer properties or other types of interesting pharmacological outputs. And then we're building machine learning models to couple that functional information, the data sets generated here, back to the computer, so we can have this sort of self-learning platform for almost real-time molecular discovery. This is just a retrospective study that we run in lab, just to illustrate how novel this field is. This is looking at PubMed results or publication results at the intersection between antibiotics and AI, cancer therapeutics and AI, and drugs and AI. And we can see that in the case of the antibiotics field, it's that intersection is incredibly young, only a couple years old. And this is just to basically emphasize that anybody in the audience, any young scientist, we really need you, to join in this effort, because we really can use your creativity, your energy, your enthusiasm, and your passion to try to come up with solutions to the antibiotic resistance problem. And so moving the next couple of slides, I'm gonna shift gears a little bit. Instead of talking about antibiotic discovery, I'm gonna talk about our more recent development of diagnostic technologies, mostly for COVID-19. And I'll tell you a bit of the backstory here. So before the pandemic, we were working on developing diagnostic tools mostly for bacterial infections. But in March of 2020, as you very well know, everything changed, and we felt this sense of professional responsibility to try to contribute something to the COVID pandemic, since we routinely work with infectious diseases. And so we decided to pivot a little bit and apply some of the technologies that we had developed to detect SARS-CoV-2. And we early on identified two primary things that we thought were important that were lacking at the time. The first one was diagnostic that was low cost, right? That everybody could afford, not only people with money, but also people that live in low resource settings, on developing countries, or in communities that maybe you don't have the monetary capacity to buy something that is very expensive. So we wanted to do that, and we focused on that. And the second aspect that we wanted to focus on, we wanted to develop a technology that was that had a very short detection time, so a very rapid technology that allowed us to detect the virus quickly. And the reason for this is that there's a lot of proof that high frequency testing is one of the major tools we have for preventing the spread of the disease, for preventing outbreaks, for preventing hospitalization, and eventually for preventing deaths. And you can only do high frequency testing if you have a rapid test. And so that was those were the two things that we focused on. So can we develop diagnostics for everybody, right? That was the concept. And again, this is just to illustrate the health inequities that have been heightened tremendously during the pandemic. I mean, they have always, of course, been there, but during the pandemic, we have seen these things explode, and to levels where, you know, there are many, many countries where not even 3% of people have received the first dose of the vaccine. Early on, when the vaccines were deployed, it was 10 countries that purchased 75% of all the vaccine doses. And so really huge health inequities. And we really felt strongly about this, and we wanted to try to do something about it. And so what we've done over the last, year and a half is we developed 3 different prototypes that I'm going to tell you briefly about. The first one is rapid, the second one is lead, and the third one is color. So I'll start with rapid. So this is a technology that is it's compatible with your laptop. It's also compatible with your cell phone. It's a low-cost test, so each one costs $4.67 to manufacture in my lab, with very little automation, so pretty rudimentary methods. And here's how it works. You can use, again, the time of detection is also very good. It's 4 minutes, so it's rapid. So again, we achieved 2 of the metrics that we wanted to achieve: low cost and rapid detection time. You can use saliva samples or nasopharyngeal or oropharyngeal samples. And the way that it works is you have an electrode and you functionalize it. We functionalize the electrode with ACE2. ACE2 is the natural receptor of the virus in our own bodies. So when the virus enters our body, there are many, many of our cells that have this ACE2 receptor. And the virus basically initiates the infectious process by binding to ACE2. So we basically recapitulated this whole natural process onto this electrode, this chip. We functionalize ACE2 here. And so when there's a sample here containing the virus, the spike protein, which is the major antigen of the virus on the surface of the virus, will bind to ACE2 and will be able to capture the binding between those 2 molecules. This is the limit of detection. So it's the analytical sensitivity is incredibly low. We can detect minute amounts of the virus in samples. And you can visualize this using an app on your cell phone, so you can discriminate between healthy and infected samples. This is just to illustrate how we build these chips. So we have, again, the electrode. Of course, you need an anchoring agent, right, to anchor the ACE2 recombinant protein. So we use glyceraldehyde, and then we add the ACE2. The ACE2 gets anchored onto the chip. And then we need 2 more steps. So a stabilizing agent, we use bovine serum albumin to stabilize the whole complex. And then we need a permeable membrane, which we use Nafion 1%. And this is mostly to prevent other things from binding to the electrode that may otherwise interfere with the detection process. This is the clinical performance of Rapid 1.0, the first prototype, again, using nasopharyngeal and oropharyngeal swabs in viral transfer medium and saliva samples. Here's the sensitivity, which basically gives you the positive correlation. So a sample that is positive for COVID or for SARS-CoV-2, do we detect it as positive? Specificity is a negative correlation, so it's the reverse. A sample that does not contain SARS-CoV-2, do we detect it accurately as negative? And then accuracy is the combination of both metrics. And so as we can see, the test is not perfect. There are some false positives and some false negatives, but generally it performs quite well. And also of note, we were able to identify some of the emerging variants. Here, when we published this paper, we had only access to the UK variant B117, but since we've done a lot of work with Delta variants as well, that are a lot more widespread at the moment throughout the globe. We also run cross-reactivity assays to make sure that the assay is specific towards SARS-CoV-2 and it doesn't, detect other coronaviruses or non-coronavirus strains like influenza strains, for example. We've done work with H1N1, H3N2, N2, etc., as we can see here. And we didn't see any major cross-reactivity events, which was exciting. And here on the right, we can see the cost to detection time matrix. Again, the 2 major things that we wanted to contribute to was low cost and low detection time. And here we can see how rapid compares with respect to other, existing tests that are FDA approved for use here in the US. Okay, so that's rapid. Moving on, I'd like to briefly tell you about lead. Here we wanted to use incorporate materials that were highly accessible. And one day looking around the lab and with my team, we realized that, of course, pencil lead graphite is a great electroconductive material, and so perhaps we could use it for this test. And so the cost for this test is $1.50. Here are some of the other parameters that are important, and it essentially looks as simple as this. You have a plastic tube with 3 pencil leads, one of them which is functionalized with ACE2 again, and then by here we can see how we use gold nanoparticles. We couple that to ACE2, and that's how we functionalize it onto the pencil lead, as we can see here. And then when the virus comes along, it basically binds to ACE2. Here we use a slightly different electrochemical approach. In rapid, we use electrochemical impedance spectroscopy. Here we use a square wave voltammetry. So in this case, a decrease in the signal actually indicates a positive signal that the virus is present in the sample. Here's how we engineered each of the graphite pencil electrodes. Again, we need a anchoring agent. This is how we add then the gold nanoparticles, and then we mobilize the ACE2 onto the gold nanoparticles, onto the graphite. And then again, we use a stabilizing agent, and we use bovine serum albumin again. Again, we've in each case, each step of the way, we've characterized each step of the way electrochemically. And this is a limit of detection. It's around 229 femtograms per milliliter. So again, we can detect very small amounts of the virus or purified spike protein in samples. We've run cross-reactivity assays. Remember, here we see a decrease in the signal when the virus is present in the sample. Interestingly, the UK variant actually gave us a stronger signal than the wild-type SARS-CoV-2 strain. And this was quite curious because, and then at the time, we also saw publications being released showing that the spike protein from the UK variant actually bound more effectively to ACE2. So this was actually aligned very well with the results that we obtained here. But we didn't observe any other cross-reactivity effects with H1N1. We can see that there is no decrease in the signal with influenza B or herpes virus or MHB, which is basically mirroring hepatitis virus. It's a coronavirus as well. And again, doing the matrix comparison of cost to detection time of lead compared to other methods, it basically tells us that lead is performing at a high level, taking into account those 2 variables. In terms of clinical sensitivity, again, we use nasopharyngeal swabs. Again, these are like cotton swabs, basically, and saliva samples. And of course, these are samples, all clinical samples from hospitals, from the hospital here at the university, and they have all been previously tested for RT-PCR. So we, of course, know, which ones are positives, which ones are negatives. And here, again, the sensitivity in each case, specificity and accuracy. I have to note here that it's 100% for saliva in each case, but please note that we only tested 10 samples. So I think as you increase the number of the sample size, that number is going to go down. There's going to be mistakes in terms of false positives and false negatives that we're trying to address at the moment. Okay, and finally, I'd like to mention just briefly color. This is the third technology. It's actually different. It doesn't use electrochemistry. It uses nanotechnology, and it's a colorimetric assay or optodiagnostic. This is almost the analogy that I like to use. It's sort of like a pregnancy test where you see a color shift when you get your positive result. Here, it operates in the same way. The really cool thing is that it's incredibly cheap. So it's 15 cents per test. And the way we've done it is we've engineered cotton swabs that are functionalized with ACE2 again, and also with gold nanoparticles, as I indicate in the slide. And when the virus is present in a given sample, we introduce the cotton swab. what happens is our system creates a sandwich with the virus, and that through a plasmonic effect, which is physically mediated, leads to a color shift, and we can detect a color shift. The limit of detection is not as sensitive as the previous methods because it doesn't use electrochemistry, it uses colorimetry and nanotechnology, but it's still quite sensitive. 0.15 picograms per milliliter to detect the purified spike protein of the virus. And it's a colorimetric assay, so you see a color shift on the cotton swab, but you can also quantitative. We've been using this RGB free app that allows you to quantify pixel intensity, so you can actually get actual data from the color shifts that you get when the virus is present in a given sample. And to the extent that actually those color shifts, the relative intensity that we get correlates quite well with CT values that are obtained from the gold standard, from RT-PCR. So they correlate very well in terms of high viral load, medium viral load, and low viral load. So that was also quite exciting to see that positive correlation. We also ran cross-reactivity assays to make sure that color did not detect other viruses that we didn't want to detect. We only wanted to detect SARS-CoV-2. And we ran reproducibility assays to make sure that when we manufactured the different batches of color, that they performed at a similar level. And we generally saw good reproducibility, despite the fact that we were doing this manually. We didn't have any automation underlying any of the manufacturing in the lab. And this is the clinical performance. In this case, because they're cotton swabs, color is a cotton swab. We only use nasopharyngeal swabs. We did a total of 100 clinical samples. And again, here's the sensitivity, the specificity, and accuracy. So again, we're making some mistakes, as we can see, but generally, the tests work well in our hands. And I think it's important to also emphasize that we don't need, the tests are probably never going to be 100% accurate. And I think that's fine as long as you make a technology that is sufficiently low cost, because then you can take the test several times in a row and rapid, right? So you can take the test several times in a row. If you get the same result 3 times in a row, let's say, even if it's 90% accurate, if you take the test 3 times in a row and it gives you the same result, that's basically going to be, if you model that mathematically, is essentially going to be 100%. And so, we don't have to be so strict about having things that are perfect, because perfection is almost impossible to achieve with tests like this. But if you have something that is sufficiently low cost and rapid, I think you can overcome that. And with that, I'd like to thank my lab. It's really a privilege to work with these people every single day. I learn a ton from them. They actually come from all sorts of backgrounds. We have people in the lab that come from physics, from chemistry, from computer science, from microbiology, from peptide chemistry. And so it's really an honor to learn from them every single day. I'd like to thank our funders for allowing us to do the work we do. And here I'm just leaving a link to our website, my email address, and Twitter handle in case anybody wants to get in touch. I always love talking science. And finally, I'd like to thank you for your attention. Thank you for attending this talk, and I'd love to take any questions that you may have.
Thank you, Professor. It was an excellent lecture. We have some questions in the chat, and if you allow me, I will read them to you.
Sure, please do.
What about the legal questions? How many % this represent in the costs of drug development? In the beginning of your presentation, you speak about this this % of the development, okay? And yeah. The student is questioning about the legal questions of this and the % that this represent in the costs.
Yeah, I mean, that's a great question. So legal associated costs add also a lot to the total cost of developing a drug, of course. So the 2.6 billion number that I put there, it includes everything. If you look only at the discovery process, that typically takes, it doesn't take the 10 years, right? The discovery process in the first like 4 or 5 years, let's say, of that process, of the entirety of the 10 years. And the cost would be, of course, less than that, but it's still substantial and it still takes years, right? With computational approaches, you can compress time, and instead of having to wait 5 years to discover something, you can do it in a matter of, a time scale of probably months. And so that's kind of how we look at it. But definitely legal fees and all that add a lot to the total cost of developing, any drug.
Okay, thanks, Professor. Another question for Samila. Congratulations, yep. Professor Cesar, for the work and research done. Very interesting and inspiring. I would like to know about the test's perspectives and if they are already available for the community.
Yeah, great question, Samila. Well, they're not approved yet for use, so they're just at this point prototypes. We are looking so we've patented the technology. We're looking into potential licensing agreements, and we're also doing pilot studies in different places. So that's as far as we've gotten at this point. But so they're not approved for use in the population yet.
Okay, thanks. My question. Using the artificial intelligence and the algorithms, is it possible to predict a resistance to a peptide, for example? If the bacteria came resistance of peptide, for example.
Yeah, that's a great question. It's something that we have not been able to model yet. So resistance development assays take a long time to do. It's 30 days, and they're very expensive. But it is a question that we're interested in. If you could, because we do see that bacteria develop, have a higher propensity of developing resistance to certain sequences versus others. So there probably must be a way of exploiting that to try to find what are the sequence determinants that make it easier or harder for a bacterium to develop resistance. And then, at least conceptually, you could think of a future where if we have enough data from those assays, you may be able to model whether a given sequence will, whether a particular bacterium will develop resistance to a given sequence. So yeah, that's something that I think in the future we may be able to do. At this point, we can't really.
Okay, thanks. Thanks again for the lecture. And
I have question also.
Octavio, go for it.
Yes. Do not stop yet. Just a little one. Because
no.
Not allowed.
Not allowed.
In fact, I have many questions, but I will keep in one or two. The first one is because we always try to improve the antimicrobial activity, and then we use artificial intelligence and other strategies, and then we improve the helix and then bind into the membranes and explode the cells, and it works. But at the same time, we just improved also the side effects. So we just start to kill the mammalian cells. So what do you think that is necessary to, at the same time, improve the antibacterial activity and decrease the unspecific activity against mammalian cells? What's the idea?
Yeah, Octavio, that's a difficult question. I mean, we are trying to build data sets and models to counter-select for toxicity or hemolysis or undesired effects. At this point, we don't have anything completed yet, but I hope that in the future we can model all of that. We can build algorithms that not only, like you say, not only potentiate antimicrobial activity, but also counter-select or remove sequences that are highly toxic against human cells or hemolytic or things that you don't really want when you're thinking about developing a drug that humans hopefully one day will use. So we're trying to build data sets we're doing experiments to build data sets to then build computational models to be able to predict that. And that's the point where we are at this point in time.
And the second one is because I really enjoy your talk. It was very nice. We show that not only the development of a new antimicrobial peptide, but also materials that can be used by functionalization of the peptides.
Yeah.
But one thing that I always I was warning is because when we put the peptides in the surface, for example, we just fixed the peptides, and then we lost a lot of movimentation and entrance in the membrane. So
yeah.
How could you avoid that? Because I saw that you show a lot of proteins in the surface, and I think that is a great idea. But how could you think that you can reduce the effect of the surface?
Yeah, I think it's still a huge challenge in the field. Like Octavio is mentioning, like whenever you functionalize a surface with a peptide, the peptide loses some antimicrobial activity because it's not as flexible. And so, one way of doing that is you can crowd the surface more. I think usually you're always going to lose some activity, right? It's not going to be as effective as a peptide that is free in solution. So we don't really have a good strategy yet to overcome that. I think you're always going to lose some functionality. I think we need to be comfortable with that unless we come up with a completely new strategy of really conjugating or functionalizing the peptides onto different surfaces. I don't have the answer to that yet. Maybe someday.
Yes, because I think that we are in the same point. So I'm always trying to get some tips from you. And just before Flavia just finishing, I need to tell to the students, this guy is one of the great guys of our field. If you want to know anything about antimicrobial peptides, I met Cesar maybe almost 10 years ago, and now he's a famous guy. So it's very nice to see him like a bright professor. He was a student, and now he was one of the best, in my opinion, in the world in this field. So thanks a lot, man, to be here with the students.
Octavio, I learned from the best. So.
Come on.
No, but yeah, no, it's really been an honor to share this time with all of you, and thank you so much for the invitation. I hope next time it can be in person and I can interact more with the students. And again, to the young audience, the students in the audience, please consider joining this effort. We really need, like I mentioned earlier, we really need your creativity, your energy, your passion to help change the world. So please consider that, and we'll welcome you with wide arms into this field.
Thanks, man.
Thank you, Professor, again for this excellent lecture. It's inspiration for all of us young students that want this area. Thanks, Dr. Otávio, for the presence too. And thanks for all to see the lecture.
Great. Thank you, everybody. Take good care, everyone. Okay. Obrigado.
Bye-bye.
Obrigado.